let's actually learn how the muscle contracts. So we're going to be going through the whole big picture, the 4 steps that are required for the muscle cell to contract. We're going to have events at the neuromuscular junction, which is going to include the release of acetylcholine from the axon terminal, which is going to start the influx of sodium, which is going to lead to muscle fiber excitation, which is going to include generating action potential, which is going to travel across the sarcolemma, across the cell membrane, and then we have excitation contraction coupling, where we're going to have that excitation, the action potential, spread down the T tubules, which is going to trigger the release of calcium from the sarcoplasmic reticulum. The release of calcium from the sarcoplasmic reticulum is going to trigger cross-bridge cycling, that binding of actin to myosin, which is going to cause the sliding filaments and the muscle to actually contract. So, at the neuromuscular junction, we're going to have an action potential traveling down the axon of the nerve cell. That signal is going to originate in the brain, go down the spinal cord, travel along a somatic motor nerve until it gets all the way to our muscle cell. So that action potential eventually arrives at the axon terminal. At the axon terminal, it's actually going to open up voltage-gated calcium channels. The voltage-gated calcium channels are going to cause calcium to move inside of the axon terminal, and that calcium is going to trigger these synaptic vesicles full of acetylcholine, that neurotransmitter, to fuse to the cell membrane of the axon terminal, release the acetylcholine into the synaptic cleft. The acetylcholine is going to bind to acetylcholine receptors. The acetylcholine receptors, which are chemically gated sodium channels, are going to open up. Sodium is going to move into the muscle cell. That is going to form what we call an end plate potential by depolarizing, making less negative the muscle cell, and that's what's going to trigger the action potential in the muscle cell. And in order to stop that signal, we have to get rid of the acetylcholine, and that's going to be done by an enzyme called acetylcholine esterase, which stops that signal by breaking down acetylcholine. So let's go through this one more time. So the action potential is going to arrive at the axon terminal of the motor neuron. Remember, an action potential is a change in voltage, a depolarization of the cell. That is going to trigger the opening of voltage-gated calcium channels in the axon terminal. The calcium moving into the axon terminal is going to trigger the release of acetylcholine into the synaptic cleft. The acetylcholine is going to bind to acetylcholine receptors on the sarcolemma. The acetylcholine receptors are chemically gated sodium channels, so sodium is going to move into the muscle cell. In order to stop the signal and stop the influx of sodium, we are going to have the enzyme acetylcholine come in, break down the acetylcholine, and cause these channels to close back up. If we have some sort of issue with either the release of acetylcholine or the reception of acetylcholine, that's going to interfere with the ability of neurons to send signals to the muscles. One example of this is myasthenia gravis. This is a degenerative disease that is characterized by drooping under the eyelids, difficulty swallowing and talking, both things done by your muscles, and overall muscle weakness. Myasthenia gravis is caused when antibodies bind to the acetylcholine receptors, blocking them and preventing them from functioning. So even though you are releasing acetylcholine, the muscle cell isn't receiving that signal and it's not opening up those channels. Because this is caused by the person's own antibodies attacking their bodies, this is an autoimmune disease. So, once that acetylcholine binds, it's going to, remember, cause an influx of sodium, which is going to depolarize the cell. So what we mean by that is the resting sarcolemma, the resting membrane of the muscle cell, is polarized. When we say polarized, we mean one side is different from the other side. In this case, what we mean is that there's a voltage across the membrane, and the inside of the cell membrane is negative compared to the outside. Its resting membrane potential is approximately negative 90 millivolts. Action potentials are going to be changes in the electrical potential across the membrane. And it's going to occur in 3 steps. What we call generation of the end plate potential. That's going to occur at the neuromuscular junction due to the release of acetylcholine. Then we have depolarization that's going to spread all across the cell. That's going to be what we call the action potential. And then we are going to have repolarization, where we're going to restore the resting membrane potential so that this muscle cell can respond to another signal. So our end plate potential, the end plate is another term for the neuromuscular junction. This is going to occur when acetylcholine binds to acetylcholine receptors on the sarcolemma. Those acetylcholine receptors are, again, chemically gated ion channels that are going to open up and allow sodium to diffuse into the muscle fiber. Because sodium is positive and the inside of the cell is negative, the positive charge cancels out some of the negative charge and makes it less negative. So it's less different from the outside. So we call that depolarization. And this localized depolarization at the neuromuscular junction is called the end plate potential. If that end plate potential is big enough, it's going to generate an action potential. Whether or not it's big enough is going to depend on the amount of acetylcholine released. If enough acetylcholine is released, causing enough chemically gated ion channels to open up, causing enough sodium to diffuse into the cell that it reaches what's called threshold, that is then going to trigger the opening of voltage-gated sodium channels. So these are sodium channels that open or close depending not on the release of neurotransmitters, but depending on what the membrane potential is. So if enough sodium goes into the cell to move the resting membrane potential up to threshold, depolarizes it enough, then these voltage-gated sodium channels open. Once these happen, we get an action potential. It's a binary. Either we reach threshold and we get an action potential, or we don't reach threshold and we don't get an action potential. But once we've reached threshold and these voltage-gated ion channels open, it is happening. We are going to get an action potential and the muscle is going to contract. Once we reach threshold and these voltage-gated ion channels open, more sodium comes in, more of the cell depolarizes. That's going to cause more voltage-gated sodium channels to open. They're going to cause more sodium to come in, which is going to cause more depolarization, which is going to open up more voltage-gated sodium channels, which is going to cause more sodium to come in, and that action potential is going to spread across the entire cell. So it's going to spread this depolarization across the whole cell. Once we've depolarized, then the cell is going to need to repolarize in order to be able to contract again. This repolarization is going to involve the closing of the voltage-gated sodium channels and the opening of voltage-gated potassium channels. Remember, potassium is at a high concentration inside of the cell. So if you open up potassium channels, potassium wants to go out of the cell. Also, at this point, the cell is no longer negative on the inside. We don't have that negative charge holding onto the potassium. It's actually a little bit positive. It's actually going to be pushing potassium out. So we again have two forces moving an ion, an electrochemical gradient. We have the concentration of potassium pushing potassium out of the cell, and potassium has a positive charge. Losing a positive charge is like gaining a negative charge. So as the potassium with a positive charge leaves the cell, that's going to make the cell again more negative, repolarizing it. This is going to be during what we call a refractory period. So the cell won't be able to actually contract again until after this refractory period ends. Because until that point, these potassium channels are open and the sodium channels are closed. Only once the cell repolarizes are the potassium channels going to close and the sodium channels going to be able to be opened again. So the muscle cell won't be able to contract again until after repolarization. So we have acetylcholine released at the neuromuscular junction, causes an influx of sodium, causing the end plate potential. If the end plate potential reaches threshold, voltage-gated sodium channels open. Once that happens, we are going to have an action potential. The influx of sodium causes more voltage-gated sodium channels to open and spreads the action potential across the whole cell, eventually triggering contraction. That is depolarization. In order for the cell to be able to contract again and receive a signal again, we are going to have repolarization, where we are going to close the voltage-gated sodium channels, open the voltage-gated potassium channels. Potassium leaves the cell, making the cell negative again. So let's look at that. So again, we have our negative 90 resting membrane potential. Acetylcholine is going to trigger chemically gated sodium channels to open. If enough of those open, to allow in enough sodium that we reach threshold, voltage-gated sodium channels open, we get an action potential in order to what we call depolarization. In order to repolarize, those sodium channels close, the potassium channels open, and we repolarize.